Right, so thank you guys. Aundigro, I'm the owner of Aundigro, and we are focusing on soil improving ingredients. We are certifying them organic if possible, and we certainly sticking to educating ourselves and exploring the natural ingredients for agriculture, regenerative farming, horticulture, and vertical farming, seeing in what manner we can improve this to the best of our abilities, sticking to the most natural ingredients that we can source. This particular webinar, we're going to run through the ingredients of our new product, Champion. So Champion is a combination of C4L Technology. I've got the technical manager from BioRevolution in this webinar. He'll be doing a specific talk on C4L Tech, which is a flavonoid and amino acid mixture. Very, very exciting. It's got just a library of academic papers written on it through the just detailing the benefits of the patent. It's an incredible product. Then we're going through protein hydrolysate, seaweed extracts, organic acids, and fulvic acids, and how they benefit soil health as well as plant health. So just to give you a little overview in the beginning, what is a biostimulant? So last year, the law came into place that registration was required for biostimulants. And according to the European Union Fertilizing Products Regulation, a plant biostimulant is a product that stimulates plant nutrition processes independently of its nutrient content. So the nutrient content will be your basic macro and micronutrients, so NPK values. So if there's anything, if all things being the same, if you're able to improve growth, then that constituent or that ingredient could be classified as a biostimulant. So it's with the aim of improving one or more of the following characteristics: availability of confined nutrients in the soil and rhizosphere. So that's making the nutrients that are in the soil more available to the plant. Nutrient use efficiency. So are you able to get the same kind of build and growth using less nutrients, and therefore the plant's being more efficient with the nutrient it has available? Tolerance to abiotic stresses, and this could also be biotic stresses, but the abiotic stresses are quite easily measurable. So under stress, under freezing, under drought, under salinity issues, pH fluctuations, those abiotic stresses are measurable. And if you can in any way reduce that, then it can be classified in that spectrum. And of course, the outcome of your crop quality, which you could measure with the Brix sugar content or cell wall strength, its ability to stay healthy for longer periods of time. And then we've got another category, which is the soil improvers. So let me just go through the definition first. So products mixed with soil or applied to the surface to enhance properties and boost plant growth. Improve soil physical, chemical, and biological aspects, enhancing structure, water retention, nutrient availability, and microbial activity for better plant growth. The composition is diverse materials, including compost, peat, organics, minerals, tailored to specific properties. The composition, it's not restricted to these ingredients. These are just probably the most common ones. But what we've done actually, because the barrier to entry with soil improvers is lower than it is with biostimulants, we first classify a new product under the soil improvement registration. And then because it takes longer in terms of trial data, we would go for the biostimulant registration following the soil improver registration. Basically, what you're doing here is, under this registration, is can you improve that rhizosphere bacterial biomass? And can you improve the activity in the soil, the structure of the soil, and the ability for there is a lot of crossover between the biostimulants and soil improvers, because you are talking about improvement of nutrient absorption, for instance, but the classifications are different. So these are the two that are new in terms of fertilizer registrations. Just one more thing, there is a chat box, so and also feel free to the champion product. It's actually certified for organic farming as well. So this means we can go right into organic farming, regenerative farming, but also just for anyone farming, you're able to know that the product makeup is of the highest quality, and it's really healthy for your soil and plants. So again, like I outlined earlier, these are the 5 ingredients. The citric acid, we've actually got an array of acids because of the protein hydrolysate in the product. So it's not just citric acid, but we do add citric acid, particularly because it not only is it helpful for the crib cycle, for instance, and for pH regulation, but it also helps to preserve the product itself. Protein hydrolysate, the kelp seaweed. This is a cold pressed kelp seaweed from Clonia maxima species. We've got the fulvic acid, and then, of course, like I mentioned earlier, the C4L technology, which Johan will talk about later. So let's go into it. It's the protein hydrolysate. Protein hydrolysate is basically the richest nutrient source that we have that's available to us outside of anything synthetic or mined. So if you're looking for a natural means to feed soil and plants, protein hydrolysate is the way to go. There's 2 different types. You can get a plant-based protein hydrolysate, and you can get fish hydrolysate or hydrolysate, depending on how you pronounce it. So we're using in our one at the moment, it's a fish hydrolysate. We are actually exploring using the legume protein hydrolysate as well, but for now we use the fish hydrolysate. And the constituents, it's got macronutrients, particularly the highest source is nitrogen. It's got micronutrients, the full spectrum, all the amino acids, so all 18 of them. And then it's got an enzyme base, because making hydrolysate, there's an enzymatic phase which takes a couple of days, and then you have the fermentation stage, which can take up to about 4 weeks. So you have the enzymes, and then you've got a bacterial breakdown phase. It's also got growth hormones. It also synthesizes growth hormones. It's a wonderful chelator because of all the acids that it has. Of course, it's got organic matter, beneficial microbes, and it's a pH regulator. Okay, so in terms of the macronutrients, it's got the NPK. I would say nitrogen is certainly the most prominent. You're looking at between 2% and 8% nitrogen, depending on the length of time that it's fermented and how it's filtered. And of course, nitrogen promotes leafy growth, phosphorus supports the root development and flowering, and potassium enhances overall plant health and diseases. The hydrolysate also contains a variety of essential micronutrients like calcium, magnesium, sulfur, iron, zinc, copper, manganese, and boron. What's really important to know as well is not only are you getting these micronutrients, but these micronutrients are chelated. So they're a lot more available to plants than it would be if you were just applying your standard format of copper or zinc. Just to give a brief overview, I know I'm probably speaking to people that are well aware of the value of these micronutrients, but I'll go through them quickly. Iron, essential for chlorophyll production and involved in electron transfer process. Zinc is required for enzyme activity, hormone regulation, and DNA synthesis. Manganese involved in photosynthesis, enzyme activation, and the breakdown of carbohydrates. So very important for breaking down your lignin and various, just your organic compounds in the soil. Copper necessary for enzyme activity and the production of plant pigments. Boron important for cell wall formation. So I haven't got it here, but boron and calcium are fantastic products to apply together because both of them are improving the vascular system and cell wall generation and strengthening, along with obviously the wonderful silica. So, and then chlorine, which is involved in osmotic regulation, photosynthesis, and ion balance, and molybdenum, which is required for nitrogen fixation and enzyme activity. There's the two products, or the two ingredients in the products. There's protein hydrolysate, but there's also the kelp seaweed we use in the Clonia maxima. Both of them have the full spectrum of amino acids. And so just focusing with the protein hydrolysate, these are ones that I've outlined. The proline, which maintains a cell structure during water stress. This is really what's important in soils, is when you do have stresses, you see the biggest difference in contribution of these products when your plant or your crop is under stress. Under really healthy conditions, using amino acids or the protein hydrolysates, you might only see a 6% difference in your yield or in measurable outcomes. But under stressful conditions, you start to see radical difference between the soils that are enriched with amino acids and the ones that aren't, as much as 25% difference in crop yields. So it may make significant differences when there's drought, freezing, any kind of pH issues, structural issues, salinity issues, too sandy soils, those kind of stresses. In sports turf, constant mowing, for instance, a lot of trampling and compaction. That's where you see really big differences. So alanine aids in nitrogen metabolism and energy supply. So again, it not only is it involved with the metabolism, but it's the transport of nitrogen. So when you've got these amino acids, you start to get far greater utilization out of the macronutrients that you have available to you. Glycine and glutamic acid, both of these, not only do they help with oxidative stress that I've mentioned in the presentation, but these two amino acids they help for the absorption of calcium and the transportation of calcium ions. This is probably one of the most fundamental contributions to plant growth and vegetative growth. So very, very important. And then fish hydrolysate has enzymes. So enzymes are basically tools. They're like a key that opens a door, but they can open 1,000 doors per second. Now, having an enzyme in your ground, it's just basically catalyzing reactions. So, and just so you understand how enzymes are broken down in words, you always have an A's at the end, so it's the ASE, and then before the A's so you'll have this A's over here, and then before the A's is basically what it breaks down. So here you've got protease, and it's breaking down proteins. Lipolysis, which is lipids, basically, right? You get peroxidase, and that's peroxides it breaks down. So that's what these enzymatic reactions are going to do, and it's filled with enzymes. So this is really something that you do want to have in your soils. So your organic acids. Now, organic acids are basically chelating tools, chelating properties. Citric acid, which we have a fair amount in the product, very, very helpful for binding to metal ions like iron, zinc, and manganese. You've got the malic acid forming complexes to remain soluble in soil solution. So also very, very helpful. Acetic acids, participates in chelating by binding to metal ions, and then you've got various other organic acids as well in the complex. Citric acid is by far the greatest contribution in our champion product, but in general, I think citric acid is the predominant acid used for chelation and for chelating your micronutrients. So when you're purchasing a product that says it's chelated iron, largely you'd find that it's using citric acid to do that. Organic matter, there's proteins, there's carbohydrates, there's fatty acids, enzyme humic and fulvic acids, and microbial byproducts. So all of these, there's a constituent that's actually of organic structure and organic compounds that you're actually feeding the ground with. There's quite a lot to go through, so I might be going a little fast through these slides, but I think I just want to, without going into, sort of an academic lecture here, I just want to give you an overview of the value of these properties. And I'm really excited actually to speak further, or let Johan speak further about the C4L Tech, which is really something quite unique. So I'm going to give him the stage at the end of this. So excuse me if I am running a little fast, but okay. So beneficial microbes. This is something that's quite particular to a specific kind of protein hydrolysate. So you get various products out there, of which some of them may contain the cultures, they may contain the spores, and they may contain various amounts of either one. So when you hydrolyze fish, what they do is they take all the offcuts of prepared fish. So that would be tail, heads, inner bones. And from all of that gets emulsified, or it gets crushed or grinded up. Then they'll break that down with enzymes. That takes a couple of days. And following that, they will they'll ferment it, and the fermentation can take it can take anything from, I think, 2 weeks to 6 weeks. So I'd say on average a month. The whole process basically takes about 2 months. Now, in that process, depending on the enzyme, the bacteria that they are using to culture and to ferment and to break down, that will also make a big difference on what comes out at the end. Now, bacillus, this is probably the most popular one known at the moment, especially in sports turf, because it helps to break down your lignin, and or your organic matter in the soil. And thatch is a big problem in sports turf. So that's the top layer of organics. It can get really spongy and can prevent a nice surface. So having bacillus is really important, and can make a big difference to improving that top layer of the profile. Then you've got rhizobacteria. Rhizobacteria, that's the bacteria around or establishing around the root system. It's called the rhizosphere. Very, very important to have a healthy culture of rhizobacteria, because they're responsible for taking the sugars that are made from the plant through photosynthesis. As exudates, they leave the root system. They're exchanged with that rhizobacteria for the various nutrients that the plants needs. So there's a bartering system between the rhizobacteria around the root system and the plant exchanging sugars for micronutrients. And as that becomes really healthy and robust, it also acts as a very protective area where the unwanted or non-beneficial bacteria can't actually get in, because it's protected by this biomass. So very important to enrich that. Not only is there a constituent of rhizobacteria in the product, but it's also a wonderful, this is probably the best feed that you can give rhizobacteria, fish hydrolysate. So then your mycorrhizal fungi, basically, in short, this is an extension of the root system. fungi is able to transport micronutrients, particularly from miles away. Well, maybe not miles, but certainly very, very far away, hundreds of meters away from a root system. It basically becomes a massive extension of the root system. So being able to improve mycorrhizal fungi volume or mass in the soil is very important. That's done where you have really good structure, soil structure, and lots of aeration. Right, and then we've got your trichoderma species of fungi. This is very important for preventing pathogenic outbreaks. It will slow down the growth of pathogens by breaking down the sort of pecking at the cell walls of pathogens. It sort of defeats them in that manner. And then the antinomycetes bacteria, that's an antibiotic, or just basically you've got beneficial bacteria and non-beneficial bacteria, and they'll compete for area. And the Azospirillum species, this is fantastic for nitrogen fixing. Nitrogen fixing means it takes nitrogen from the atmosphere as opposed to taking nitrogen from organic matter or from fertilizers, for instance. So it can actually take the NO2, the nitric oxide, which is in the air, and convert that into ammonia and then make it available to the plant. So also fantastic product to have when you want to reduce your fertilizer regime. It's a pH regulator. So fish hydrolysate, because of all the acids that it has, amino acids, humic acids, all the other organic acids we mentioned earlier, they act as buffering compounds. They don't necessarily change the pH of the soil, but what they will do is regulate spikes, so you won't get big changes or fluctuations in pH. And that's very important for maintaining a healthy biomass, because biomass can be quite sensitive to pH changes. Okay, so that's it on the protein hydrolysase. Does anyone have any questions for me? I see in the chat. Right, I'm going to do a certainly a shorter overview of Aclonia maxima Kelp Seaweed, because I had, the last webinar was entirely focused on this ingredient or this plant. So I'm going to quickly go over this now, just because it still is a constituent. Gentlemen, the pH level of the fish hydrolysate is around 3.2, if I'm not mistaken. It's between 3 and 3.5. I don't recall the EC value. There is obviously a salinity, or there is a sulfur content, but that will be available. I think it's actually available on the website. I just don't know it offhand, but I can get that back to you. All right. Quick overview of the most important attributes. Polysaccharides, very important. Betanes and sterols. The betanes and sterols help for the strengthening of not only the cell walls, but also the ability that it's like an osmoprotectant, the ability to transfer nutrients equally and protect them within the cells. So the betanes and sterols help for the elasticity of the cell walls and the structure of those cell walls. Growth hormones, it has a constituent of growth hormones, but more importantly, it synthesizes growth hormones within the plant. Sugar alcohols, osmoprotectants, and antibiotics. Oxidants. Sorry. The mannitol is probably, I've just highlighted it here because what you find when people are looking these days for a really good seaweed extract, they want to see what the measurement of mannitol is. And this has become quite a popular attribute to seaweeds. And that's mannitol's an alcohol, and it acts as an antioxidant. Essential amino acids, of which there's essential and they've got the full spectrum. Phenols that are actually the antioxidants here. This mannitol should maybe even be in this section. And then, of course, the macro and micronutrients. I did mention this earlier. So let me just go through the ones that I'm highlighting here from the kelp extracts. Glutamic acid and glycine, these I would say are actually the most important, and they just make such an incredible contribution to the vegetative growth and to the strength of the plant. So it stimulates root cells to open calcium ion channels, allowing plants to take in calcium ions thousands to millions of times faster than simple osmosis. So this makes an incredible contribution to vegetative growth and to the health of the plant through simply having these amino acids available. Arginine plays a vital role in nitrogen storage. Proline helps with stress. Tryptophan is a precursor to the growth hormone auxins. Methionine is a precursor to ethylene, and also which is also going to be a precursor to growth hormones, playing a vital role in plant growth and development. So there's the three plant hormones that are available in kelp, but not only available in kelp, also they are synthesized through the amino acid contents of kelp and the polysaccharides. It's auxins, which are responsible for the growth and elongation of the cells. And then you'll get cytokinins, which is for cytokinesis. That's chopping them like scissors in half. So it will grow tall through the auxins, and then when they grow big enough, it will chop them in half with the cytokinins. And you get gibberellins, and gibberellins are predominantly used for the germination, and sort of initial growth. Phenols and fluorotannins and antioxidants, that's your colorants as well. This is falling under the metabolite section. So this helps tremendously for stress tolerance, and that's what I was talking about earlier. This is where we see the biggest differences in trial work between the control and the treated areas. As under stress, we see major differences in being able to withstand stressful times. So it mitigates oxidative biotic stress, pathogen protection, antimicrobial properties, suppress soil-borne pathogens, and nutrient uptake, stimulates root enzyme and enhances nutrients absorption, detoxifies, neutralizes pollutants and toxins in soil, and then, of course, the water efficiency, increases plant water use efficiency. Here's the full array of, sorry, we just gotta, here is the full array of metabolites and enzymes. Within the kelp, you will find that, and we're going to talk further because there are a small amount of flavonoids, but we're largely looking at your vitamins and minerals. And then you'll have the carotenoids in the middle here and your polyphenols. The flavonoids particularly found in the C4L later. We'll go through that a little later. And then you get your enzymes, some of which we actually source from other plant materials, like peroxidase, for instance, which is a fantastic enzyme for releasing oxygen. Forms of extraction. Here we've got, this is for the kelp, enzymatic extraction. The downside is that alkaline acids are used, and this can break down some of the valuable properties. Fermentation, similar story, it destroys some of the growth hormones available. Cold pressing, this is the one that we source. So cold press is basically just squashing out the liquid. That's what you want. So kelp is 85% liquid, and all the goodies are inside that cell walls. So using a cold pressing machine to get those liquids out is the best, and then filtering out the solids. Micronization, fine milling. This is also very valuable because you're getting basically all the fibers, so really good for mixing in your soils. But then you do, of course, lose a lot of the liquids. Right. Just to tell you, basically, this is an industry that's growing incredibly fast. And to give you an idea, it's generating total value of 500 billion globally. This is just seaweed alone. And sorry, no, that's not true. That's biostimulants. Um, and it's all used for the productivity in agriculture. So this is quite amazing that, and these are the various different seaweeds here. And these are the totals just generated per region. This is the region where we're getting our Eclonia maxima. It's from up the west coast, just north of Cape Town. There was a meta-analysis that was done, and the meta-analysis was done on the full spectrum of biostimulants over 1,000 pairs of open field data in a total of 180 qualified studies worldwide. The results, this is a very brief summary. The add-on yield benefit among all biostimulant categories is on average 17.9% and reached the highest potential via soil treatment. Biostimulant applied in arid climates and vegetable cultivation had the highest impact on crop yield. So that's what I was mentioning earlier, arid climates, that would be a stressful constituent. And then biostimulants were more efficient in low soil organic matter content, non-neutral, saline, nutrient insufficient, and sandy soils. This is available on the UCOP website if you want to get the meta-analysis. Fantastic read. Right. Just looking now at the difference between synthetic amino acids and, synthetic auxins and using Eclonia maxima, you can see there's a difference in the two, and by having a natural source, you always get a better outcome. We had to spend a bit of time to find out what the optimal rates would be. And, so with each crop, there's a different rate, although we have narrowed it down to, in general, under not too much stress, the 3 liters per hectare is most often used under most conditions. We see this is another one just expressing the difference between a stressful environment, you're getting a 25% difference, and optimal environments, only a 6% difference. Right, let's go through fulvic acid. Now, fulvic acid, just explain to you what fulvic and humic acids are. Your soil, originally your leaves and grasses and, dead mammals and insects, they're all dying, and as they degrade, they start to form the organic content of the soil. And over years and years and years, this develops into a very rich sludge or a rich compound soils, and then eventually it gets compacted and compacted and compacted into fulvic and humic acid, or what they call humus. So the humic acids are the smaller particles, and the humic acids are the larger particles. If we didn't extract this, for instance, out of lenodite, then when it gets compacted further and further, it becomes coal. So it's basically the stage before coal that we're extracting this, and it's incredibly valuable from a nutrient content or its carbon content, but it's got these additional benefits. Nutrients availability and uptake. It's because it chelates. Fulvic acid has the ability to chelate or bound positively charged ions and make them available to the root system. So by putting fulvic acid into your soils, what happens is the nutrients that are binding to the soil particles, they get chelated by the fulvic acid, and because of the charge differentiation, they are able to move the micronutrients from the soil particle, the clay particles or the sandy particles, through to the root system and make them available to the root system. So it makes a massive difference in terms of nutrient uptake. Promotes nutrient absorption due to its small molecular weight. Fulvic acid can penetrate plant cells, enhancing the uptake of nutrients like magnesium, calcium, and zinc. Root growth development stimulates root elongation. Fulvic acids can stimulate root elongation and branching, leading to a more extensive root system. A robust root system allows for better water and nutrient uptake, enhancing plant resilience during stress conditions. So what they've also shown through studies is that by combining your fulvic and humic acids with amino acids from your protein hydrolysate, you get a 1+1=3 situation. In both cases, the product on its own, it's when they're used together, you get far greater reach, or the results are measurably improved. So the benefits extend further into the promotion of beneficial microbial activity. Fulvic acid can stimulate the growth and activity of beneficial soil microbes. These microbes play a role in nutrient cycling, improving soil structure, and suppressing soil-borne diseases. It improves soil aggregation. Oh, sorry, there might be another person that wants to admit. There we go. Improve soil aggregation. So aggregation is just getting a really good particle size for soil, allowing for good aeration between particles. By promoting fungal growth, especially mycorrhizal fungi, fulvic acid can improve soil aggregation, better soil structure, enhancing water infiltration, root penetration, and reduces soil erosion. It also has antioxidant activity. Fulvic acid can mitigate oxidative stress in plants by scavenging reactive oxygen species. This antioxidant activity can help plant cope with stresses like drought, high salinity, and extreme temperatures. Stimulates enzyme activity. Fulvic acid can enhance the activity of various enzymes in plants, aiding in metabolic processes and improving overall plant health. It also supports chlorophyll production, and of course, leading to enhanced photosynthesis. This improves the ability to create sugars, which is the source of all life. So being able to benefit the soil and the plant with better chlorophyll production is incredibly important. It binds toxins as well. In polluted environments, fulvic acid can bind to heavy metals and various toxins, reducing their availability to plants and mitigating the harmful effects. Right. Just before I pass on to Johan, I'm going to quickly have a look at the questions. I see there's quite a number of questions. Maybe I should just leave these. Okay, well, thank you very, very much for all your questions. I'll tell you what's best, just so that we consider it to everyone listening. I will answer all these questions at the end, and we can have a bit of a Q&A. And for those that want to depart out of the meeting, obviously they can. It's now 3:45, and I'll pass it over to Johan. Please introduce yourself, Johan, and then if you can just go through the details of C4L, that'll be great.
Hi guys, welcome to all the online viewers. Thanks, Luke, for the mini introduction. I'm the technical and business development manager of BioRevolution, and we are the owners and the manufacturer of C4L Technology. I've been involved with C4L Technology for around 15 years, especially in a technical capacity. I was part of the development process and most of the research that was done. So we are pressed for time. Luke said he's going to give me a few extra minutes, Luke. It's going to be tight. Perfect. So maybe just off the bat, I think to mention that we are really just going to skim the surface here today. This will be an introduction, almost a teaser to what will follow in a full seminar in probably six weeks time. So to start off, what is C4L technology? As you can read with me, it's a carefully formulated complex of unique flavonoids and different plant extracts, including citrus aurantium and organic acid. So it's a mixture of 4 different extracts, and citrus aurantium makes out the biggest component of the extracts. I think what is important here to mention, it's stating the obvious, but a lot of the success of the product lies within the formulation process. Because it is extracts, it's a very delicate mixing procedure. We have about 9 steps that we go through, and the sequence of these steps are important. If we mix it up, we see a lot of problems with separating in terms of the product. Also not adding too much heat, otherwise you can damage some of the biochemical compounds and their bioavailability as well as their structure. So the formulation is important. The ratios between the extracts, the type of extracts that are used. So not all bioflavonoid containing products are necessarily equally made. Important to mention is that it is an organic product, serious certification as well as OMRI. It's a suspension concentrate, excellent compatibility. We've been using it for more than 20 years in different situation tank mixes, in can mixtures. We haven't come across any problems with regard to that. So not a solution, but a suspension concentrate. So once you apply, it goes out of suspension and it's available to be taken up. Like I've mentioned, extensively tested for more than 20 years and on more than 48 different crops. So we pride ourselves that we are a data-driven company, and C4L Technology is a data-driven product, and it is very crop-specific when it comes to recommendations. Just a little bit of background is in the early days when I was involved, 15 to 20 years, we started with a lot of trials in the field. So randomized small plot field trials as well as demonstration trials, and playing around with dosage rates for various crops and timings and intervals. And as we went along, we saw just amazing results, from yield increase to size increase, coloring improvements, shelf life, things like that. But it's almost a reverse engineering. Many years back, there was a growing desire to figure out what is going on in the plant. Why are we seeing what we are seeing? We had an interest to see the mode of action. And that led us to various studies at university level, where we did honors degree and MSc Masters studies as well as PhD study. And from those studies, we got to see some of the mode of action, what is at play, what is going on within the plant. And that helped us to connect the dots. So almost a reverse engineering, like I've mentioned. And here it is. So today we're going to have a very quick look at the mode of action. It is really just surface level stuff today. We're not going to take a deep dive, and hopefully this will be a teaser for what is to come. So what does C4L Technology do? It enhances and supports plant and soil health by optimizing and enhancing natural plant processes to increase production, resilience, and product quality. So within this PhD study that I'm referring to, we saw after the application of C4L, there was an activation of more than 2,500 genes in the model plant that was used, that was Arabidopsis. So just for starters, at the real basic level, after the application of C4L technology, there was a major response within the plant. And we'll go into what that response was. But it seems like when we look at the mode of action, it starts with enhanced photosynthesis. And this, of course, leads to increased plant energy. Now, plant energy is very important to mention here. Is almost like the currency in a plant. For us, it would be like money, maybe in worldly terms. If you have more of it, you can do more things. And similar to plants or having a vehicle, if you have a vehicle, you have potential to drive, to go places, but you need fuel in your vehicle. Otherwise, it is just potential. So similarly, plants has many mechanisms to work with, but for a start, there needs to be enough energy. So that's an important component. Then moving on to increased root exudate. So if we have enhanced photosynthesis, we know that a large portion ranging from 5 to 21%, some literature says even 40%. And if you have mycorrhizal colonization, it can even increase. But that's a large portion of the photosynthesis that is exuded into the rhizosphere. And this has a major impact on the rhizosphere microbial community. And I think we will get into that a bit later. There's major positive effects that we see. So the rhizosphere that you will be aware of is that small thin layer of soil around the root. It's about 1 to 2 millimeters, and it's directly impacted by the root. And of course, some of the root exudates, or most of the root exudates, carbon compounds, but it is not only carbohydrates, it is proteins, it's enzymes, it's secondary metabolites, and all these things have various functions and advantages in the soil environment. From there, we move on to the pathway. So there's an inducing and suppressing of various pathways in a plant. Ultimately, it shifts the plant towards secondary metabolism or a stress priming response. So there's signaling involved, hormone regulation, the production of secondary metabolites, and then a broad priming response. Again, just to pause here for a moment, not to go into much depth, but the secondary metabolism is very taxing to the plant when it comes to energy. So here we go back to our energy. If we have enough energy, we can make use or proper use of this secondary metabolism that has many benefits, especially the secondary metabolites when it comes to things like pollination, UV protection, chemical defense systems, detoxification, and they can also be used in the plant as antioxidants. And maybe for the next session, we will go into much greater depth the role of secondary metabolites within a plant. Luke, thanks. Let's go on to the next slide. So here is just a teaser of the PhD study. This is we call it a heat map or a gene map that was drawn up by the student. And just in short, we see red and we see green, and we see intensity varieties there. So this is, the green is upregulation of genes, red is downregulation of genes, and then we see the intensity. So it's not just on or off. There are intensity levels of being upregulated as well as downregulated. We'll get into the detail. Just for now, I want you to have a look at the top right corner there. There's a block just below light reactions, and there's a lot of green going on there. So that's the upregulation of genes involved with photosynthesis. So when we say we enhance photosynthesis, we see it now by the reverse engineering onto a genetic level, that we have upregulation in genes involved in photosynthesis. All right, just remember that there's a lot more, but we don't have time to do it now. So I want you to move on, please, Luke. So here is the example I want to use. So when we keep your mind with photosynthesis just for a moment, and the upregulation of those genes and all the green that you saw. So when we see the upregulation, so I think there was 15 genes specifically involved in chlorophyll synthesis that was upregulated. So that would mean if we go on and measure chlorophyll content, there should be a difference. So this is the first linking of the dot. So the left top corner, you'll see the control plant. Left bottom corner, that is a C4L applied plant. It's a 0.01%. It's a very low rate. The top right corner, that is a salt stress situation. So the PhD study was based on a salt stress situation. So as an example of abiotic stress. So just to mention before I forget, one of the first things that's affected in a plant when it comes to stress, and specifically abiotic stress, is chlorophyll and chlorophyll content, as well as photosynthesis. Then when we look at the top right, it's a 50 millimole is a moderate salt stress situation. In the study, which we will look at next time, there's also 100 millimole sodium chloride that will be more of a salt shock situation. But we can see that the biomass is affected by the stress situation. Clearly, we can also see the yellowing, the chlorosis that is coming through. Now, the bottom right of that picture, you'll see that's the same salt stress situation with 50 millimole with C4L at the very low rate that is applied. And visibly, it's different to that of the salt stress alone, but it's very equal to that of the control. So biomass-wise, as well as the greening of the leaves. So we move over to the actual chlorophyll content measured in this Arabidopsis, that is the model plant that was used. So the control is in red. Next to that will be C4L. So the control there and the C4L, that is a no stress situation. All right. Then we see the statistical significant difference in the chlorophyll content that is visible in the picture as well, but also difference in the measured chlorophyll content. The black block will be the lower one, is the sodium chloride 50 millimole. So we see the impact of the salt stress situation on chlorophyll content immediately. It's statistically lower than the control as well as the C4L alone. But right next to it, you see where I'm going. It's the sodium chloride 50 millimole plus the C4L at 0.01%. And it helps the plant tolerate the salt stress situation to the extent that it's the same as the control, as the unstressed plant. And that is really remarkable. So this is the first link that I want to show you. Just from a genetic level, the genes expressed or upregulated chlorophyll synthesis, so the production of chlorophyll. When we link the next dot, it will be here, where we see it measured in the leaves. We also see a visible difference. And then we go all the way, when you follow photosynthesis, to the many, many trials on a lot of different crops that we have in the field situation. The dots keep on linking. We see size differences, we see yield increase, and many other benefits that I can give you. Right, the next and the last slide will be just pointing out some of the benefits that we see. So like we spoke of just now, there was a salt stress or an abiotic stress situation, but we see improved abiotic and biotic stress tolerance through the induced resistance and as well as stress metabolism. We'll get into that next time. So the stress metabolism, like cell wall metabolism that is upregulated, means that it's a strengthening and a thickening of the cell wall. And then there's a lot to say about secondary metabolites, but they play a major role when it comes to a stress response. So we have the abiotic and the biotic stress, the ones mentioned there: salinity, drought, heat. We have results in the field situation, measured results, trial results on all of that I've mentioned, as well as biotic stress, pest and disease. So examples will be, that comes to mind, is powdery mildew, xanthomonas, that will be a bacterial disease, scab on pome fruit, fusarium, that will be a soil-borne disease, and we'll get into detail at a later stage. When it comes to soil and root health, major impact on the soil health, and that predominantly comes from that root exudate. So we have an enhanced root development. Important, the root exudates, that's increased, like we've spoke of. And then the activity, the increase in the activity of beneficial soil organisms. we see improved colonization on the roots. Now there, I want to point out, we have data there on different crops when we see the measured difference in the colonization of mycorrhizae, for example, on roots, on wheats, on kiwis, on grapes. We see an increase in colonization, but also in the structural formation. So the arbuscular mycorrhizae that is increased. So the nutrient exchange mechanism that is developed further where C4L is applied. Now we know a major role player. I need to not go into the rabbit holes here, Luke, but a major role player when it comes to root exudates, when it comes to symbiotic associations, it is secondary metabolites. So when you increase the secondary metabolites in the root exudates, they play a major role in forming these symbiotic associations with your beneficial mycorrhizae and other bacteria, which we also have studies for. It's actually university studies that we did that. The suppressor of harmful soil organisms. So here we have it. We are impacting the rhizosphere positively with the increase of the root exudate. So over time, we see actually a shift in the population of nematodes. We've seen a shift, a downward or a decrease of parasitic, the harmful nematodes, and an increase in saprophytic nematodes. It's just a teaser, so I won't go into much detail, but there's a lot of crops that we've tested that we have the results for that. And then also with that positive impact, we see the soil water holding capacity being improved, as well as soil penetrability. And then going on to the crops, we improve yield, seed germination, flower stimulation, fruit set and retention. This is very crop specific. We have a lot of data on crop specific data when it comes to all these things that I mentioned. It's measurable, and we've done very many trials over many years and still continuing. I just had results last week of citrus, where we improved the flower initiation and stimulation. And so it's a continuous thing for us as a company. We see fruit size. You remember the energy that I've mentioned, the increase there, but also not only size, it's also size distribution. And that goes from vegetables all the way to your tree crops. We see a size distribution shift. So less of the small ones, less of the bigger ones, they're all pushed into the to the desired sizes. Then fruit color, as I mentioned previously, fruit finish, it's wide range, but it's russeting, cracking, things like that. Nutrient content, I just want to point this out here, for example, just to explain or give an example of the many trials that we have done and the vast data set when it comes to nutrient content, and then specifically calcium. We have more than 30 trials done on 10 different crops, and we see an average increase of calcium of 27%. Important to mention is that the product itself doesn't contain calcium. So it's an improvement in the soil health and the rhizosphere, and of course the tree health. Then fruit firmness, another area. We have a lot of data, as well as shelf life. And I think I'm gonna stop there, Luke. Otherwise, we're gonna continue for a long time. But this is, like I've mentioned, only a teaser, and hopefully we can take a deep dive in a few weeks' time.
Thank you so much, Johan. That was fantastic. Even for a teaser, that was quite a deep dive. I think some of the jargon used is not familiar to everyone in the audience, so it's really fantastic to have you on. I think we will sort of vaguely plan for a 45-minute to an hour session focused on Crop for Life tech in mid-November. So that'll be very exciting. And the wonderful thing about C4L is they've been trialing for decades now. So they have a wealth of experience in every different crop type. So particularly in agriculture, So any of the farmers or agronomists that are in the audience, that'll be a fantastic one to follow. And then for sports turf as well, just knowing that the C4L is involved in the champion product, it's just fantastic to know that you're getting, all these benefits. So thank you very much, Johan. Guys, this is now the end. I see we've gone over time. It's now 8 minutes past 4. So I would completely understand if you want to drop off. There are a number of questions that have been asked, so I'm going to run through them. So if you want to stick around for the Q&A, please do, and I'll quickly run through them. Johan, there's a couple of questions for you as well. So let me just go through them. But listen, thank you for everyone that spent, over an hour listening to us. It's really wonderful to know that there's such a great interest. I think there was about 50 people in the audience. So thank you very much. I really do appreciate your time, and I hope this was valuable to you. If you've got any questions for us, please feel free to just send me an email. If you have interest in the product Champion for your own use, please let us know. We'd love to get you out some product. So drop me an email to luke@aundigro.com. You can also just drop it through the website at aundigro.com. You can drop a question. And I think a lot of you have come through to us through LinkedIn and following the Aundigro LinkedIn page. So you can also DM me there. I have so many people actually on there. I think there's anyway, there's a number of messages on there. So either email luke@aundigro.com or just directly through DM. Right. In what form, this is from Gabino, forgive me if I don't pronounce your names correctly, but in what form do you add citric acid? It's in a liquid form, and it's added at the end of the composition for, like I say, just to increase the chelating ability and to preserve the product. Gentlemen, what do we have here? What is the pH? Okay, I spoke about that earlier. The pH is just over 3. And I don't have the EC value for you right now, but you can, and it's easy to get that for you. There is a salt content, so there will be an EC value for sure. What is the volume based between hydrolysate and seaweed extract? So the composition is 55% protein hydrolysate. It's 21% Econia maxima kelp seaweed, 21% fulvic acid. And then the rest will be C4L Technology and citric acid. That was from Luke Ansel. Thank you, Luke. Great question. Andreas, is that correct? Do you add some microorganisms into the fish hydrolysates? It seems that some bacteria, rhizobium and mycorrhiza fungi are specific for soil environments, and they need plants for some symbiotic relationship development. You're 100% correct, and that's why when I went through that, it's not that there's the constituent of those bacteria within the hydrolysate, it's that it promotes the growth of those bacteria and fungi. Albeit there are some spores that you would find in the liquid itself. The cultures won't be available, as you highlight, because the cultures need the environment of soil to grow. And then Anna Bravo, how is it ensured that the amino acids keep their function and structure after it has been processed? Well, you're able to actually measure the amino acid content in our products following the process. So we're able to, in laboratories, analyze the amino acid content, of which we have done numerous times, and there's always the full availability of those amino acids in their full structure. So the amino acids in the fermentation or the enzymatic breakdown process, they are breaking down the proteins into amino acids. So in some cases, you can take it too far where it would even break down the amino acids themselves. But in the case of, well, maybe more so with the kelp, there's absolutely no processing. It's just cold pressed. So there you would have the raw amino acid content. But there's a very rich availability through the hydrolyzing process of a rich sources of amino acids that maintain a good structure. Right, Hamza, how do you obtain kelp? If you harvest it from forests, what kind of measure are you taking towards making a sustainable product? The Eclonia maxima that we harvest is under strict regulation. So, for instance, there are two different measures you can take to harvest from forests. You are only allowed to harvest once every 6 months from a designated block, and that's only if you are harvesting from the surface. If you are diving and you want to take from the anchor, then the holdfast, then you can only harvest once every 5 years if you're taking down the full leaf, the full plant. So those are the two regulatory requirements. But as you can imagine, for harvesters themselves, it's not only in the interest of keeping the environment in a healthy condition, for the harvesters themselves to maintain a crop that's available to harvest. You don't want to destroy the area that you've been designated. So you get given blocks to harvest that are for you alone. You get a license for an area, as if you were buying property on land. You get licensed for an area, and then that area is regulated. And it's, and not only is it regulated, but it's also in the interest of the farmer to manage it well, so that he's always got healthy crops to harvest. Now, just so that you're aware, Eclonia maxima, or kelp in general, grows incredibly fast. It can grow up to 0.5 meter per day. So if you are harvesting, albeit that regulatory requirements are saying that you can only harvest once every 6 months, it actually will grow to the point where you couldn't tell that we had even harvested within a couple of weeks. So 1 month later, it's generally speaking replenished. It does the differences, seasonal differences in growth. Obviously, in the warmer temperatures, it grows a lot faster. But generally speaking, it's a plant. It's the fastest growing plant on the planet. It's like the bamboo of the sea. Hence the growth hormone stimulation. I hope that answered your question. There's also a write-up. If you go to the UKelp website, ukelp.com, then there's a write-up about the regulatory requirements and the full breakdown of the procedure as well. How can the fulvic acid chelate nutrients in the soil if it is chelating the compounds in the mixture? So that's not exactly how it works. So you have there's the chelating ability of let's say the acids, for instance, and well, the acids and the fulvic. There's a tremendous rich amount there that once it's applied to the soil, there's a transfer of not only ionic exchange, but you've got just so much material that it's certainly not all getting absorbed by the nutrients that are inside the product itself. Albeit that nutrient is of course going to be chelated, that's in the product, and that is the nutrient that you want to make available. So it is the predominant nutrient that you want to be chelated, but it's going to now be available. If you imagine just like an oil droplet, right? If you've got an oil droplet and you put a piece of salt next to it, it will chelate that salt. And if you I'm just using this for a pic, so that you get a picture. But it doesn't mean that it's not able to chelate everything else. It'll spread around whatever particles are around that droplet. So chelation works in the same kind of effect. How this is for you? This is Andreas again. Another question from is we've got how the C4L extracts are applied in soil application or by leaf spray application. Johan, this is for you.
Yeah, so is my mic on? Yes, yeah, it's still on. It is applied either foliar, oil or soil application. So a lot of the data, most of the data actually is as a soil, or rather as a foliar application. But we have data on soil applications as well. So either way, we have good results. I'll read the next one.
Yeah, from James.
So if you're claiming C4L improves biotic stress tolerance, please can you clarify why this isn't classified as a plant protection product under that code? All right, James, thanks for the question. Yeah, maybe just to circle around one word that you've mentioned there, it improves biotic stress. So in our trials that we've done, we have a objective, we have a target focus when it comes to small plot registration trials, and mostly that has to do with yield and quality aspects of fruit set or flowering and things like that. And what happened over the years, different crops, we will see a pest or a disease come into the trial area. Of course, you have a untreated and you have C4L applied. What we do throughout the trials is we'll keep the pest spray programs, pest and disease spray programs. Of course, we'll keep it the same throughout the trial area. But then we'll see, that was about 20 years ago, we started to see, especially when it comes to the biotic stress, as you've mentioned, we see differences in powdery mildew and mealybug and thrips and nematodes when it comes to the soil. And at the different trial sites that we've done over the years, we've seen for sure improvements, but we are actually in the process. It's still early days in moving towards what you have suggested, but there's a difference between improving biotic stress and to control a pest of a disease. And for us, it's sometimes a vague area. So what is improvement? Is it 30%, 40%, 50%, or is it 80 or 90%? So there's a lot of gray areas to work through still for us. That wasn't the aim. In any case, we are aiming for a biostimulant, and we have the results on the abiotic stress side measured in the field as well, like I've mentioned, but then also biotic, something that people not always mention, or haven't come across a lot of biostimulants mentioning the biotic stress component. So the moment through the registration process, I don't want to make it too long, but if you want to claim a specific pest or disease, of course you have to replicate that for many times in different climatic zones, and if you want to, if you're comfortable then on various crops, then you go for that registration. So I hope that that brings some clarity to your question. But I can mention that we are in the process for us with C4L Technology to explore, actually, when it comes to the soil part, we are exploring for a registration, as you've mentioned.
Yeah, perfect. Thanks for that. And I mean, there's one of the things about being a plant protection product and the registration. It's an incredible registration to go for. In terms, it takes an incredible amount of time, and it's massively expensive. So we would certainly initially, when it's an introductory product, we would remain on its ability to enhance the plant's ability to defend itself far more than us saying that this particular chemical or combination of chemicals will control a disease.
Yeah, look,
Johan, there's another question there from Adriage. Hydrolysate. I hope
Yeah,
I'm pronouncing it correctly.
referring to the PhD study on the sodium chloride stress. Did you test different concentrations of C4L? Did you test other abiotic stress factors? So the PhD study is a massive study. It's I think it was 6 or 7 years, and you can't make it too broad. So we worked on Arabidopsis. It's a model plant, but then we also worked on Solanum lycopersicum, which is the tomatoes. And yes, we did a different rate on the tomatoes, and that will be in, if I can call it, the deeper dive. I will show that data to you as well. But and in that situation, it wasn't a stress situation. That was a hydroponic grown situation. But we only tested the sodium chloride, and we had to fix the rate. It was too difficult. Even for that, you can imagine the student had to sift through more than he thought he would have in terms of gene activation. So that was the focus of the study, was on a molecular and a genetic level, what is going on in the plant. So when it comes to the field situation and field trials and very crop specific recommendations, we've tested field trials throughout the years to see where the sweet spot is in terms of the dosage, the intervals, the number of applications, as well as the timing when to apply. Okay.
There will be another presentation that we're going to give on where we're focusing our attention on the value of monosilicic acid, as well as going through the various silica products that are out there. We have a product called Gladiator, which is monosilicic acid with boron and calcium, and that'll be the discussion will be kind of around that. Thank you again to everyone. Really appreciate you being in our audience. Take care and have a lovely evening further.